It really is a wonderful pleasure to be here on this special occasion, so thank you, Sunny. Okay, we've been hearing this morning about this structure, and it sort of never fails to amaze me that how much of life boils down to these relatively simple interactions with three hydrogen bonds connecting a C and G to form a base pair, and T and A. So this section is really about methodologies for determining the primary sequence of DNA. And I entered this field somewhat by accident, but one of the tenets for sequencing and for the Human Genome Project was the fundamental nature of DNA and its sequence towards understanding all living systems, and in particular understanding the genetic basis for what makes us who we are and the underlying causes of disease states. Of course, we tend to focus on humans. There are many organisms on the planet that have DNA that form their genomes. So all of these points apply across organisms. Oops. Now, there was a lot of talk about sociology of practicing science with other human beings, and the importance of that, and that's relevant to my story because I bumped into sequencing somewhat by accident. Many years ago, at the start of my career in Cambridge, in the chemistry department, I was doing some experiments and I needed a laser. And my background is organic chemistry, and I learned about the kinetics and the mechanism of DNA polymerases from my time as a postdoc with Stephen Benkovic in Pennsylvania State in USA. And I was introduced to this character, David Klenneman, who was a physical chemist, a laser spectroscopist, who had trained in Dick Zayer's lab at Stanford. And we carried out the experiment I needed, and then we kept talking, and we decided to write a grant to do a project that combined my interest then on DNA polymerases and his interest on single molecule spectroscopy. And we had two postdocs. Mine was an organic chemist, Colin Barnes, and he had a postdoc, Mark Osborne, who's a physical chemist. And the experiments that we were carrying out were fundamental in nature. We were absolutely not trying to do anything particularly useful other than learn about the mechanics of nucleotide incorporation by a DNA polymerase. And so part of the setup that we built was a single molecule TIRF microscope. And we were doing experiments in various formats, and one was to take a fused silica surface and immobilize a synthetic DNA template carrying a primer on it, and to bind a DNA polymerase to that DNA and watch the enzyme incorporate building blocks one at a time in this template-directed synthesis by the polymerase. Now, there were a number of different ways that we approached this problem, but we needed an observable, we needed a fluorophore. And so one of the ways we did this was to actually incorporate a fluorophore into the deoxynucleotide triphosphate in order to incorporate fluorescently labeled nucleotides. So these were the experiments we were carrying out. So I'm going to introduce you to another meeting house in Cambridge. If any of you have visited the chemistry department or in fact the engineering department, this is a place we gather. The chemistry department is just here. You can see the roof of it. And actually, in my early days as an academic, I used to spend a lot of time here because we didn't really have proper offices. So if you wanted to meet someone and discuss science, discuss experiments, troubleshoot things that weren't working, we would often just go around the corner and chat in here. And this is myself and David Klenneman. And in '97, we were actually thinking about, we were diverted from our experiments. We were aware of the Human Genome Project taking place at the Sanger Institute down the road. And here we had discussions about really subverting what we were doing towards this. Taking our immobilized template primer system, if you could color code each of the 4 building blocks in a way that signified the identity of the base, the thought experiment was, we should be able to incorporate the correct Watson-Crick cognate base pair. So here you'd incorporate an A opposite a T. If we could persuade the polymerase to incorporate these modified building blocks. And second part is if we could stop the reaction at that point qualitatively by single molecule imaging of the surface, we could read off this color and that base and get one base of information. Then using chemistry, if we could remove this fluorophore and protecting group, we could go here into the N+1 position and then incorporate the next nucleotide, which would be this T opposite this A, bringing with it this color. So sequentially, have cycles of color changes that give us the primary sequence of DNA. Now, in a sense, this only gets you solid phase sequencing of one DNA fragment. But the advantage of this is the potential to parallelize massively simply by extracting genomic DNA. One can fragment with restriction enzymes or by shear forces mechanically. And we did experiments early on that showed us that we could monodisperse fragments onto our surface in a way that could be imaged and also interrogated enzymatically. So each fragment sits in its own optical resolvable space. Now, if you apply solid phase fluorescence sequencing to this, you're able to get information from all of these fragments simultaneously and achieve speed and capacity by massive parallelization. So we did a very simple back of the envelope type calculation in '97. We made some assumptions turned out to be incorrect, other assumptions turned out to be an underestimate of what was possible. But taking an array size, the kinetics of single nucleotide incorporation by a polymerase, we calculated that it would be possible to build a system that could sequence on a scale of billions of bases per day or a few days. That was the thought experiment. And these early proof of concept experiments led us to form Solexa in 1998. Now, for me, this is arguably one of the most important chemical reactions in life. It's the reaction catalyzed generally by polymerases, which is the nucleotide transfer reaction, whereby one nucleotide is added to the terminus of a growing strand of DNA. So this 3' hydroxyl here being extended corresponds to this position. And what a polymerase generally does is it has two acidic residues that help bind two magnesium ions when the triphosphate comes in. And the triphosphate is an activated nucleotide. And then you get nucleophilic attack for the chemists who are here from this 3' oxygen onto the alpha phosphorus. And this part is activated by the first magnesium. The second magnesium binds to the beta-gamma phosphates and lowers the pK of the diphosphate leaving group. So this is an inline attack with inversion at phosphorus. So we had to re-engineer the chemistry of this reaction and the polymerases. And Fred Sanger's approach lacked this oxygen, the dideoxy approach. And so the nucleotides he used irreversibly terminated synthesis to generate a ladder of DNA that terminated at one of each of the 4 bases. And by sizing the DNA, you could determine the sequence. We set out to incorporate what chemists call a protecting group, but the sequencing community have tended to call this a reversible terminator by analogy with the Sanger approach. So this needs to be removable when we choose to do so. And then we need a fluorophore that can be attached through mechanics that allow it to be removed, but also allow it to be incorporated by the polymerase, whilst preserving the Watson-Crick hydrogen bonding interactions that are important for the accuracy of DNA synthesis by a polymerase. And this can be done by modifying the non-Watson-Crick edge of bases. So this is a T base, so you can modify this position for T and also C. For G and A, we actually substitute N7 for a carbon, and then we attach the fluorophore there. It was really key to get the size and characteristics of this protecting group right, and also to use chemistry that was compatible in water, chemistry that would not cross-react with other parts of the DNA, so chemists often refer to this as bio-orthogonal chemistry, and work with very, very high efficiency, because if you want to sequence DNA, you need to do N reactions, and so there's a compounding effect if it's not very efficient. Now, there's more than one chemistry that can work, but the first generation sequencing nucleotides carried this group, an azido-methyl protecting group here, and the same corresponding group here. And this bond here, this carbon-oxygen bond here, and this carbon-oxygen bond here, these are the bonds that are cleaved after each incorporation in readiness for the next cycle. Now, this chemistry is actually very old. It came out of the work of a German chemist called Hermann Staudinger in the early 1900s. Now, he discovered that phosphorus-3 reagents called phosphines are very good at reducing azides to primary amines. And if you form a primary amine connected to oxygen through an aliphatic carbon, this is called a hemiaminal. This spontaneously hydrolyzes in water to release the alcohol group. So this is a relatively small group, and we reasoned it could be accommodated by the polymerase. Now, it took a while to find polymerases that could tolerate these modifications. And I haven't got time today, but there was quite a lot of engineering that took place in Solexa, guided by the wonderful structural biology on polymerases in the literature, to mutate and optimize the polymerase for this technology. So we had set out to sequence arrays of DNA molecules by single molecule fluorescence. Then I had an encounter with Sidney Brenner, who by this time was dividing his time around the world, California, Cold Spring Harbor, Cambridge, UK, and also Singapore. And it was another one of these social encounters, actually, and he persuaded me that we should really be amplifying these single molecules to get copies in order to ensure that the sort of stochastic single molecule fluorescence events that are interesting physically but could lead to error generation would be improved by amplification. Now, again, there are a number of methods for amplifying DNA that can be used. Now, we adopted a method that was created by Pascal Meyer and his colleagues in a company he founded called Mantea Predictive Medicine. And in essence, the way the method works is you can take a fragment of DNA and use standard molecular biology and synthetic adapters to ligate up the ends, and they can be the same or they can be asymmetric adapters. And we can engraft onto the surface now of a flow cell synthetic DNA that complements the sequence of the synthetic adapters and capture the DNA on the surface by hybridization. And the way this approach, the Mantea approach, works is, in essence, it's rather like carrying out PCR, but in 2 dimensions. So you can prime one end and extend if you add a polymerase and unmodified dNTPs. And then you can melt this duplex and re-anneal, and statistically you'll re-anneal to engrafted primers on the surface, and you can extend again and undergo cycles of geometric amplification of the clonal molecule. Because one end is always anchored to the surface, these molecules remain in the same spatial location. So you generate a cluster of amplified molecules derived from the seed molecule on the surface. Now, sequencing not only improves the accuracy through having more than one vote, as Sidney Brenner used to put it, for each base, but now we no longer need what were then very expensive and also quite bulky detectors and equipment for single molecule detection, and we could use cheaper cameras. So this was the first small genome sequenced using this approach, and this is the genome of bacteriophage 174, which was also in Fred Sanger's famous paper in PNAS. This was the genome that he sequenced, and you can see here each of these spots is a cluster of molecules, and the color signifies the identity of the base in that step. Now, we reasoned early on that if we could sequence reads of at least 25 bases, we would be able to computationally realign to the reference sequence being generated by the Human Genome Project. Of course, nowadays, the chemistry and enzymology and detection efficiency is good enough to do 250 bases, and you can sequence from one end and prime and sequence from the other end to get paired 250s, and I think now you can do paired 300s quite well. Oops. So in 2006, Alexa released this system called the Genome Analyzer, and it actually achieved our calculated aspirational goal of 1 billion bases in a single experiment. Now, this technology was acquired, and the company was acquired by Illumina in early 2007, where they have continued to optimize and engineer on many levels the technology. And today, some of you will recognize this instrument. Now, this can sequence $6 trillion per experiment, or the equivalent of I think a 50x genome an hour in terms of throughput. So my last slide really is, early on, our driving force in this, because we were both chemists, was to really enable others to do population-scale whole human genome sequencing, which we thought was really needed to truly understand the genetic basis of humans. Now, there have been many large-scale projects in different countries. This is international. This is the UK, Finland, and the US, just as examples of sequencing. This project is in conjunction with the NHS, the state-funded healthcare system of the UK, and has sequenced many patients across oncology, infectious diseases, and also rare diseases. You'll hear more about this from Dennis Lowe. This is an area he has pioneered, sequencing cell-free DNA. But I'll say that, you know, as a reflection on this, population-scale human genome sequencing is, of course, here. And I believe we're really at the beginning of understanding what is possible in personalized medicine through having individualized profiles of your genetic makeup. Many people that I have to thank from the University of Cambridge, from the early days of this project. Solexa was where the whole thing was reduced to practice, and it required the talents of an incredible interdisciplinary team of scientists, and also the team at Illumina who've improved the technology. I'll leave you with these reflections. Thank you.